Hello, everyone. My name is Catalina. I'm the CEO and co-founder of OneSkin. We are a longevity company developing solutions to extend our health span, and we are starting from the skin. Skin has been primarily related to its aesthetic and beauty aspects, but it has a major role in our overall health and longevity. Most of us are familiar with the term inflammation, which refers to the chronic activation of our immune system, which further leads to several age-related diseases. Skin, being our largest organ, heavily influences our body's level of inflammation, and by keeping our skin healthy, we may be able to help preventing and delaying the onset of such chronic disease. Senescent cells are one of the main sources of inflammation, and very briefly, these are damaged cells that stop proliferating and start secreting inflammatory molecules, which further contaminates other cells around, just like bad apples in a basket. And the accumulation of senescent cells has been associated with several age-related disease and tissue aging, and skin is no different. Therefore, we are focused on developing interventions that will decrease or prevent the formation of senescent cells in order to extend the health of our skin. Similar to most interventions in the healthcare system, current solutions to promote skin rejuvenation are targeting the symptoms of aging and not aging itself. Moreover, several of those strategies are even detrimental to your skin in the long term. We are fundamentally different. We start by understanding the skin aging biology and then addressing the underlying mechanism of this process. So we have developed a comprehensive platform where we first replicated skin aging in the lab by growing human skin based on cells derived from donors of diverse ages. We were able to replicate neonatal, young, and aged skins, and we further characterized them in terms of histology, gene expression, senescent cell content, and so on. Here we can clearly see how the epidermal thickness decreases over the aging process. Next, we have developed our skin-specific molecular clock in order to measure skin biological age in vitro and in vivo. So using samples derived exclusively from skin, we have built our own algorithm that we were able to predict skin molecular age with a higher accuracy when compared to known skin-specific algorithms available. We are now using this as a tool to validate interventions so we can bring these dots above this linear regression in order to down to this area in order to achieve a lower molecular age compared to your chronological age. Next, we have built a cell-based screening platform to identify novel senotherapeutic compounds, similar to what has been published before. And we have screened more than 1,000 of compounds, and we highlighted here in blue our hits, which were able to decrease the relative number of senescent cells, but didn't decrease the total cell numbers, characterizing them mainly as xenomorphic compounds. After several rounds of optimization and validation in different donors, we got to our lead compound OS1 that has been able to reduce from 25 to 40% the level of senescent cell burden. And here we are representing by a staining with beta-galactosidase activity, which is a well-known marker for senescent cells. So now we are combining all these pieces of our platform where we first produced an aged skin that were then treated with OS1 or retinoic acid, that's considered a gold standard molecule for skin aging. And we have assessed the molecular age through the methylation profile and also the skin health score through this histological analysis. And we have seen that OS1 was able to decrease the molecular age and increase this histological score, while retinoic acid, although it decreased the molecular age to a smaller extent, it didn't increase the skin health score, mainly to its well-known side effects caused by inflammation and this peeling effect where the upper layers are coming out of the skin. So next, we proceeded to validate a topical application of OS1 in human skin biopsies, and its effect was further confirmed by increasing the thickness of the epidermal layer, here highlighted in purple, while retinoic acid was not able to promote such effect. Before moving to clinical studies, we had performed all the safety studies recommended by FDA guidelines to consumer personal care topical products, and we have assessed cell toxicity, mutagenesis, karyotyping, chromosomal aberration, and finally we also assess human sensitization through this patch testing, and we haven't found any adverse effects. So then we finally designed our clinical study with 22 patients to assess efficacy with a topical application of OS1, and after 12 weeks we could see a significant improvement in several parameters of skin health that can be clearly observed by these images acquired from one of our patients. When we correlate in vitro and in vivo data, we see a very interesting correlation where a higher epidermal thickness was also observed in an improvement in a better skin barrier on an average of 18%. Lower levels of senescent cells, inflammations, and lower biological age has been also associated with an improvement in several aspects related to skin elasticity, firmness, wrinkles, and overall skin appearance. So this makes us very confident to bring our first product to the market, a product that was scientifically designed to promote skin health from within. We have applied the rigor of a drug discovery process in order to develop a trustworthy consumer product, and we are showing that targeting senescent cells is an innovative and effective way to promote skin rejuvenation. So we would also like to explore other applications beyond the skin, and then we have assessed a lifespan study with OS1 where we could see an improvement in health span parameters with C. elegans and also an extension in the median lifespan in 12%. OS1 has also shown a promising activity in modulating inflammation when we used immune cells that were activated and then subsequently incubated with OS1, and we could see a lower proliferation of such cells, and we also could see an increase in expression of anti-inflammatory cytokines such as IL-10. So this opens new interesting applications such as psoriasis, rheumatoid arthritis, lupus, and even COVID-19, which needs to be further explored. In terms of go-to-market strategy, we are focused on bringing our first topical product to the market, delivered direct to consumer through our website. We have been working to build a science-based longevity consumer brand, and we expect to launch our product by September of this year. In parallel, we'll be exploring other longevity applications via partnerships and in licensing agreements. I would like to share a glimpse of this journey that has been both incredibly challenging and exciting. We started 4 years ago when we joined InVibio in 2016, and throughout the years we were able to build this robust platform that I shared. We raised 2 rounds of financing, we filed 3 patents, and we are now preparing to launch our product. I'm proud to introduce a scientific and female-led team that's challenging the status quo, and we gather more than 30 years of expertise in skin biology, tissue engineering, bioinformatics, and stem cell biology, and we were very fortunate to attract brilliant scientists coming from the best research groups in the aging and biotech field, and we are now growing our operation and marketing team with seasoned entrepreneurs from the beauty tech space. We are also honored to have brilliant scientific collaborations that are leading experts in the field of aging, dermatology, and cellular senescence. I have summarized here the main takeaways of my presentation, but I'll leave it here for your further reference. And I would like to invite all of you that want to redefine your limits and build a future where aging won't make us more susceptible to any disease to join us on this mission. Thank you so much.